So, photosynthesis. It's been known for a long time that the general reactions of photosynthesis require carbon dioxide from the air and require water from somewhere, and in the presence of both light and chlorophyll, CO2 and water can be converted into sugar plus molecular oxygen. This has actually been known for 250 years now. The details of this have become more clear to us over the last 50 to 70 years. And we now know that the basic reaction for, if we combine the light reactions and the dark or the carbon reactions of photosynthesis, the basic reaction takes 3 molecules of carbon dioxide plus water, again in the presence of light and chlorophyll, and this produces a sugar plus 3 molecules of oxygen plus 3 molecules of water. So a couple important things that you should know about this. First of all, the product of photosynthesis is a 3-carbon sugar. Don't let anybody tell you glucose or sucrose is the product of photosynthesis. The product of photosynthesis for any of the types of photosynthesis we'll talk about is ultimately a 3-carbon sugar. Okay? So to get that, we need 3 CO2s to go in. That's a good question. I mean, you look in any introductory biology textbook, even in the beginning of the chapter, the very first page, it says a 6-carbon sugar there. I don't know where these guys' heads are at. They go on to describe all the process of photosynthesis and never mention glucose as a product. So I don't know why that happens. It's a very good question, but it's very misleading because you want to see the triose sugars are the products of photosynthesis. And how those triose sugars then fit in to the rest of carbon metabolism is something we'll talk about next week. Okay. So the energetics for this, the delta G is on the order of plus 115 kilocalories per mole of CO2 fixed. Big energy requirement to make this reaction happen. We know this is the case because if we run the reaction in the other direction, we call that respiration. And that produces a lot of energy that basically runs all our cells. Okay, so photosynthesis, this energy requirement is obviously being met by light, and we'll see how that happens in just a minute. Okay, one of the key things that led scientists to really understand this is the net reaction. There are many, many individual reactions behind this, some of which we'll spend some time talking about, most of which we won't. But to get to this point, to understand what's going on here, one of the key things that contributed to that was the work of a guy by the name of C.B. van Niel. He was a Dutch microbiologist, and he studied photosynthesis not in plants, but in bacteria, and mostly in a very wide range of bacteria. The photosynthesis that we're going to study today, or over the next couple of lectures, that happens in higher plants, is only one mechanism for photosynthesis. There are many, many different types of photosynthesis, but most of them are in bacteria. But what C.B. van Niel came up with when he studied this was a general equation that described photosynthesis in all these different organisms that he had looked at. And this equation was NCO2, where n is some integer, plus 2n H2x. And we're not going to just specify what x is. X turned out to be a number of different things depending upon the type of bacteria he was looking at. Produces a sugar, and at that time he didn't know how many carbons were in the sugar. It was a carbohydrate with the general formula CH2O and raised to some nth power. So he didn't know how this reaction happened at first, but he knew that this equation, where you substituted lots of different things for X, one of the simplest ones is sulfur. There are bacteria that take hydrogen sulfide and oxidize that hydrogen sulfide to molecular sulfur. The obvious one that we think about is X being oxygen. So the production of molecular oxygen at the end of this. But from this general equation, C.B. Van Niel suggested a number of different things about how photosynthesis worked. At the time, his colleagues thought he was nuts. No one believed anything he was suggesting. But every single thing he predicted has turned out to be correct. And we'll just look at a few of them as we go along. Okay, so let's think about this general reaction in terms of oxidation and reduction. So remember what oxidation and reduction is? Oxidation, you're taking electrons away. Reducing, you're giving electrons to it. We'll come back to that in a minute. But what we need to recognize is that carbon dioxide is being reduced. You're adding, actually, hydrogen atoms to it to make sugars. So this is a reduction. And that H2X is being oxidized. And one of the things, well, let's take a brief digression here to think about redox reactions, what we generally mean by a redox reaction. So let's take a simple reaction where we have A with an extra electron on it, plus B going to A plus B minus. An electron transfer reaction where an electron that was on A got transferred to B. We could also generalize this as the transfer of a proton, a hydrogen atom. AH plus B goes to A plus BH. Okay? In either of these reactions, A minus or AH has given up an electron. It's been oxidized to A. And B has been reduced to either B or B minus. So one of the things that you should be comfortable with in terms of thinking about respiration and photosynthesis, nitrogen metabolism, when we talk about nitrogen assimilation in plants, there are a number of different reactions where electrons are moving around, where some things are losing electrons and other things are gaining electrons. And you just need to be fairly comfortable with the terminology, because I'm going to use it a lot. And if you don't understand the difference between oxidation and reduction, you're going to have some difficulty. Okay, so in biology, let me make some room here. There are many cases where an electron can be passed from one compound to another directly. But it's much more common that things move by electron shuttles. So a reaction we could have where A is being oxidized by a compound we'll just call NAD, and there may be a phosphate or not be a phosphate there, plus a proton. Sorry. So in this case, A minus has been oxidized by this compound, forming the reduced version, NADH or NADPH. And then in a second reaction, the electron is transferred from this compound to B, regenerating the oxidized form. So NAD or NADP and NADPH are electron shuttles that take electrons from a place where they're removed from some compound and shuttle them to other reactions where something needs to be reduced, where the electron's being given to it. So we'll see examples of both of these types of reactions today. In electron transport, photosynthetic and respiratory electron transport, these types of reactions dominate. In carbon metabolism, these types of reactions dominate, particularly in the reduction of CO2 to sugar that is accomplished by electrons that are shuttled by NADPH. Okay, so we'll see specific examples of this, but thinking about the role that redox reactions play in moving electrons around in electron transport and in the carbon reactions is going to be important. Okay, questions on that before we go on? Does oxidation and reduction reasonably comfortable with that? Because if we're not, let's take a second. No? Okay. All right. So let's go back to our reaction. The other thing you may have noticed is we have water on both sides of the equation, and somewhere back in chemistry someone told you you could cancel those out and just put 3 waters here. Well, you'll see that water's on both sides of the equation for a specific reason. Because one of the things that C.B. Van Niel suggested was that this is not a single concerted reaction, that it occurs in 2 steps. The first step is the oxidation of water. Let's see. Let's do this. Let me do this on a per CO2 basis. So 2 waters go to make molecular oxygen plus 4 electrons plus 4 protons. And the delta G for this reaction is positive. It requires an input of energy. And then the second reaction is taking the CO2 plus the 4 electrons plus the 4 protons and reducing the CO2 to carbohydrate. And the delta G for this reaction is less than 0. So what C.B. Van Niel suggested was that the overall reaction of photosynthesis that he had described was really 2 separate reactions. An energy requiring oxidation of water to produce molecular oxygen and electrons. And then using those electrons to reduce CO2 to carbohydrate. And at the time, he called this reaction the light reactions, and this reaction dark reactions. We now call it the carbon reactions, because normally this reaction does not happen in the dark. It only happens in the light, but only because it requires light to produce the electrons to make this happen. So this was very controversial. It was controversial because it suggested 2 things. First of all, these were separate reactions, not a concerted reaction. But it also suggests that the oxygen that's produced comes from water and not from carbon dioxide. And for whatever reason, there was a very strong feeling at the time that the oxygen that's produced in photosynthesis comes from carbon dioxide. So C.B. Van Niel's hypothesis was very controversial. It was subsequently showed using isotope techniques, labeling the oxygens in either water or CO2 and seeing where that label showed up, that he was correct, that it does come from molecular oxygen. Okay, so, or it does come from water. So what this means is that we can divide in a logical sense photosynthesis up into two separate groups of reactions. The light reactions, the energy requiring reactions, which produce, well, we got electrons here, but we know that there's, or we should remember that there's no such thing as free electrons in biological systems. These electrons get shuttled using that carrier, NADP, NADPH, and then they're used up here in the carbon reactions. So the energy requirement of photosynthesis, the requirement for sunlight, occurs in this first reaction. Once this first reaction is completed, the CO2 fixation reaction happens spontaneously. You've already stored the energy in compounds that allows this reaction to happen. Okay, so we can logically separate the way we study photosynthesis into two groups of reactions. The light reactions that we'll focus on today, and the carbon or the dark reactions that we'll focus on tomorrow. And, Tuesday and Thursday of next week. But just to link these together, let's ask ourselves, what is the actual units, what are the actual compounds that transfer the energy that is stored in the light reactions to the carbon fixation reactions of the dark reactions? And per CO2 fixed, for every CO2 that's fixed into carbohydrate, the minimum requirement is 3 ATP and 2 NADPH. So we know about ATP in terms of moving energy around. We talked about using it in terms of driving forces for primary active transport pumps and things like that. But we're less familiar with this. But I think I erased the reaction. 2 NADPH is equivalent to 4 electrons. So per CO2 fixed, we need 4 electrons to do that reduction reaction. So we're going to see that sort of keeping track of electrons is something that's important to do in the light reactions of photosynthesis. So we should be thinking about the mechanisms by which the light reactions convert the energy that's in sunlight. We'll talk about that energy in just a second. Convert the energy that's in sunlight into ATP and NADPH, which then can be used to drive this CO2 fixation reaction spontaneously. Okay? I'm looking out there and not quite sure that the faces indicate that you understand this. Yeah? Yeah, where does the ATP come in? We haven't even talked about that yet, and we won't talk about it until next week. So somewhere in the reactions that take CO2 and convert it into sugars, both, we can see where the electrons come in, at least in the general sense. There's no indication where ATP comes in here, but it does. There is a, if you think about it, if the delta G for this reaction, if there has to be an input of energy to make this reaction go from the electrons and from the ATP, then there's probably some step in that sequence of reactions where the delta G is positive. And that's where ATP is going to come in. And as I said, we'll see the details of that next week. So this is just sort of to put our initial focus on generating the things in the light reactions that the dark reactions need. Okay? All right. So this slide, in terms of your overall understanding of photosynthesis, is probably the most important one. A relatively simple slide that shows the separation of the reactions that C.B. Van Niel suggested. the light reactions, and the Calvin cycle, which is the dark reactions or the carbon reactions. And as C.B. Van Niel suggested, one of the things that's happening in the light reactions is the oxidation of water to produce molecular oxygen as a byproduct. But the other thing that has to happen is converting ADP to ATP and NADP to NADPH. The electrons that were taken away from water to make molecular oxygen go to NADP to make NADPH. Okay? So thinking about this in terms of the dark reactions requiring these two compounds, everybody's got that. You don't have to think about that. That's very clear. But the other side of this is equally important. The Calvin cycle uses up ATP and NADPH, and in the process regenerates ADP and NADPH. In other words, the light reactions require these compounds to be returned from the dark reactions, from the Calvin cycle. Let's take a simple scenario. Let's think about, well, first of all, let's remember that the Calvin cycle requires CO2, right? Let's take a leaf that's photosynthesizing happily in the light, and the roots run out of water, and the stomates close. Still lots of light there, ability to do lots of photosynthesis, but now CO2 can't get in because the stomates are closed. What happens to the rate of oxygen evolution when the stomate's closed? Emmy? Why?
Because the stomates are closed and they can't leave, but and so it might affect, like, the Calvin cycle might start capturing oxygen?
We will see that that can happen, but you're missing the key thing that I tried to draw your attention to from this figure. Anna, you want to take a crack at it?
Well, if the Calvin cycle was warm as well, CO2,
Okay.
you won't be able to regenerate the NADP and ADP.
Okay, so what will happen to the rate of oxygen evolution?
It will be less.
Yeah, it will go down. It will go down.
So how could the Calvin cycle be called the dark reactions when the stomates are closed at night and you're not going to have CO2 coming in?
The dark reactions is really a misnomer.
Yeah.
Because the dark reactions in any photosynthetic organism don't happen in the dark. They happen in the light because the ATP and NADPH are being produced only in the light. Okay? So if you take a chloroplast and you feed it ATP and NADPH under the right conditions, you can make it do carbon fixation in the dark. So we should really say the light-dependent reactions and the light-independent reactions, because this doesn't directly depend on light, but it does depend on the products that are made in light. But getting back to what this coupling is that Anna correctly pointed out, if the Calvin cycle slows down because there's no CO2 available, because the stomates are closed, then it stops using up ATP and NADPH, but it also stops producing NADP and ADP. And those are required by the light reactions. If the light reactions can't get those, doesn't matter how much light is coming in, they can't happen. The electrons that are being taken away from water are reducing NADP to NADPH. If there's no NADP, there's no place for those electrons to go, and you can't make any oxygen. So this coupling, remember this word we've used several times? This coupling is an obligate coupling. You can't get around it. And it is one of the most important concepts in terms of understanding how photosynthesis is regulated. That this intimate coupling between the light and dark reactions means anything you do to perturb the light reactions will affect the dark reactions. But equally, anything you do to perturb the dark reactions will affect the light reactions. So even though there's lots of light available, if there's no NADP, electron transport can't happen. You can't have oxygen evolution. You can't make any ATP. You can't do any of those things. Okay, Anna?
But ADP and the other one is available in other cells, and the other cells use ATP.
Yeah. So does ATP move from cell to cell? Yeah, it's a good question. Does ATP move from cell to cell? Francisco, you're saying no. Why?
Well, that would be too easy.
That would be too easy. Yeah. Let's think about it from an evolutionary perspective.
Well, but that's why we transport it in form of sugar or something to make energy.
Yeah, so if we're going to move energy from one cell to another, we do it in the form of sugars, not in the form of ATP. Think about it from an evolutionary perspective. In a prokaryote, a single-celled organism, is there any reason for a prokaryote to export ATP? No. No. And so that has continued along. Eukaryotic organisms, there are very few circumstances where ATP moves from cell to cell. Same for NAD and ADPH. If you're going to move energy from cell to cell, you use that energy to make sugar or an amino acid or something, and you move that compound from cell to cell, and then you convert it back on the other cell.
So the only thing that are happening in the cell, well, in the chloroplast,
This is in the chloroplast, right?
Are the light and reactions in the cell?
Yep. Well, as far as photosynthesis is
Well, that's
concerned.
what I'm saying. So are there any other processes?
Oh yeah, lots.
So why don't they, I'm sure they use ATP.
They do.
Okay, but they don't. So this is just like sequestering one part of the cell, or?
No, this is just in the chloroplast.
Okay. What I'm saying is, aren't there other processes?
Yes, there are other processes that happen, and do you think that the chloroplast transports ATP in and out?
Yeah.
Where did the chloroplast come from?
One-cell organism.
Yeah, one-cell organisms, and those one-cell organisms transport ATP or NADPH in and out. Think a chloroplast does it? It does not. It does not. You can get the energy of ATP and NADPH out of the chloroplast by using it up in the chloroplast to make sugars and then exporting the sugars out of the chloroplast into the cytoplasm and then doing respiration to get the ATP and NADPH back again. But there are no transporters for ATP and NADPH in the chloroplast. So it's a really, you're asking all the right questions. If you hadn't asked them, I would have had to tell you this. Because it doesn't make any sense if there could just be lots of ATP and NADPH exchanging with the cytoplasm. There is none. The chloroplast has a fixed pool of ATP and NADPH. And once it's all in this form, everything comes to a grinding halt because there's no ADP or NADP available for the light reactions.
But the evolutionary argument can't be the only reason, because you could ask, well, in a unicellular organism, there's no reason to transfer sugars. There's no reason to do any other thing. Is it because ATP is more reactive, or there's more chances that if you move it around that it will do things that the cell doesn't want?
That's a good question. So why sugars over ATP? Yeah. Why would it move sugars from one cell to the other but not ATP? I don't know the answer to that.
Do they think maybe the sugars is more stable in terms of particle than ATP and ADPH?
Certainly we know that some sugars are more stable than others, and those ones that are more stable are the ones that are transported. So that might have something to do with it. I mean, this is both the benefit and the problem of asking questions in the context of evolution. We'll see that there's lots of things where we say, does this make sense from the perspective of evolution? And we'll say yes. There'll be lots of questions, including one we'll talk about in a few minutes, where an evolutionary perspective gives you an answer that you wouldn't expect. And probably what that means is we're asking the wrong question. Right? We don't know enough about this to really understand it. Do we know enough about the stability of ATP versus sugars, or all the other competing reactions that might be going on to assess this in an evolutionary sense? And I think the answer is no. But nevertheless, the chloroplast envelope is completely impermeable to ATP and NADPH. So what happens inside the chloroplast is dependent upon its own pool of these compounds. And so if everything is in the form of ATP and NADPH, there's none of this, the light reactions are going to stop. If there are other reactions, as Anna suggested, that are using this up, that would allow the light reactions to go faster, right? Because there'd be a bigger sink for these products. Understanding that coupling is really important. Okay? Yeah.
Well, that's not going to give any advantage to the plant. It's not going to produce any sugar that it can.
Yeah, so if you allow this to be used for other reactions outside the chloroplast,
Yeah, or we put something in the chloroplast, whatever, we transform the plant.
yep.
Well, it's not going to give any advantage to the plant. We can have like reaction and more oxygen. That's
Yeah, and in fact, you might want to look at it the other way. It would be a disadvantage because those would be reactions that would be competing with the ability of the plant to fix CO2. So there are reactions in the chloroplast that use these compounds, but the reason that happens is because that's the only place in the plant cell that those reactions happen. So for example, the production of nucleotides, the bases are made in the chloroplast. They're not made in the cytoplasm. They're not made in the mitochondria. Hemes that are involved in electron transport, main parts of the hemes are made only in the chloroplast. So there are lots of reactions that for whatever reason in evolution are restricted to the chloroplast and only use energy, at least directly, from processes in the light reactions in the chloroplast.
Is there more ATP in chloroplasts than in other parts
No.
of the cell?
No. Yeah, so that's the other part of this question that we should be asking. How big are the pools of ATP in the chloroplast or in the cytoplasm? They're not big. They're very small. So if you look in the cytoplasm and you ask how long does it take an ATP to get converted to ADP and back to ATP again, cycling around like that? Typically 10 times a second. So it's a small pool that's cycling really quickly. It's even faster in the chloroplast. Okay, so if this pool is small, it's very easy for it to become completely in the ATP and ADPH form and have none of this available, or the other way around. What's the advantage of that? What's the advantage of having a small pool rather than a big pool? This would be a really good exam question. Okay?
It is, to make the reactions more sensitive, it's easier to regulate. Once ADP is used out.
Exactly. Exactly. It provides a mechanism to carefully regulate these things, to couple them together in some logical way. We'll see this a lot more clearly when we talk about respiration, because respiration has the exact same sort of coupling. We have a bunch of carbon reactions and we have electron transport. And the coupling between those two turns out to be very important in regulating respiration.
So I was thinking, since there isn't all that much ATP in the
Yep.
cell, like maybe the reason why like chloroplasts, for example, didn't evolve a way to transfer ATP in and out, it's because since there's so little of it, it won't diffuse much, and also like there isn't the gradient, so maybe it would have to be active transport, but because active transport uses ATP, then like how are you going to transport it? Maybe that's why.
Yeah, you could do that, or it might be from the perspective if you manipulate, if you make this pool much smaller, it could actually limit the Calvin cycle. So if something else needed ATP out in the cytoplasm, and the chloroplast gave up the ATP, that would further limit the chloroplast's ability to do photosynthesis. So let's leave your good thinking about why this is the case from an evolutionary perspective behind, but keep the focus on the fact that these compounds tightly couple the light and the dark reactions. You can't affect one without quickly affecting the other, because the pool that couples them is very small. Okay? All right. So let's get to one of the topics that a number of you asked questions about today, and that is about light and pigments and how energy is conserved and things like that. So we really want to focus on this idea of energy transduction. So if you go outside on a cool day with a white-colored shirt on versus a black shirt, do you notice the difference? Or if you park your car in the sun and your car is white versus black, do you notice the difference? Yeah, you should. the black will heat up more than the white. Say that again. Yeah, okay. So it may not happen in Ithaca because we don't get enough sun. But you have, in your own experience, you know that there's energy available in sunlight. If you go outside on a sunny day with dark-colored clothing on, you'll get warm. If you have light-colored clothing on, you won't get as warm. The difference is that the pigments in the clothing, and Anna's black sweater, or Himmy's black sweatshirt, you wear those outside, they absorb sunlight and convert that sunlight into heat energy. Right? You can put a pan of water out in the sun, and it'll evaporate. Right? That's, again, or sunlight energy that's being converted into thermal energy of molecules moving around in the atmosphere. So we know there's energy there. What we need to ask ourselves is, how do chlorophylls, the pigments, the catalytic pigments that are required for photosynthesis, how do they play a role in that energy transduction? Because remember, what we need to do is we need to take energy in the form of photons, sunlight, and convert it into energy in terms of redox reactions, oxidants and reductants that drive electron transport, that make these reactions produce ATP and NADPH so that these reactions can happen. That's the real thing we need to think about. Okay, so one of the important things to keep in mind is that we know that there's energy, but how much energy? How much energy is there in a photon? Well, a red photon at about, let's say, 650 nanometers, the red end of the spectrum. What is that? That's about 150 kilocalories per mole of photons. That's the wavelength, yeah. NM? Oh, that one? That's a Greek lambda. That's wavelength. If we take a blue photon at the other end of the visible spectrum, say at 400 nanometers, the energy here is on the order of 375 kilocalories per mole. So somewhere back in chemistry or physics or somewhere, you learned that the energy of light is inversely proportional to its wavelength. Shorter wavelength, higher energy. You know that from a practical perspective because ultraviolet light causes a lot more damage to living organisms than infrared light. Infrared is further, longer wavelength than 650. Ultraviolet is shorter wavelength than 400. Right? So ultraviolet causes damage because there's a lot more energy in those photons, and that energy can be used to cause damage. What we want to do is think about how the energy available in red and blue photons can be used to drive photosynthesis. Okay? So the first step in converting the energy of the photons into energy that can be used in photosynthesis is the process of absorption. And in the process of absorption, we need to keep in mind the first law of thermodynamics, that the energy of the photon must be conserved. You can't make energy disappear. The energy of the photon must be conserved through the absorption process. So absorption requires not just the photon, but something to do the absorption. We'll just call it a pigment. Pigment P plus a photon. What happens to P when it absorbs a photon in terms of energy? It's got to be higher energy, right? So we'll call this higher energy photon. We'll give it a little asterisk there. And we'll call this P star an excited state. So what's happened is the energy of the photon has been conserved in a higher energy configuration of the pigment. What's happening here is easy to understand. You remember from somewhere back in chemistry about putting electrons into orbitals? Remember you had the orbitals and you put electrons with their arrows pointing in opposite directions to fill up the orbitals, but then there's more orbitals up here that are empty. What happens when a molecule absorbs a photon is one of these electrons that are out here in the most distant orbital moves up to one of the higher orbitals. If we think about that strictly from an electrical perspective, we got a nucleus that's got a positive charge in it, and we've got electrons in these orbitals with negative charges in it. Electrons are negative. If we're going to take an electron and move it from this orbital to this orbital, we're pulling positive and negative charges apart. Does that take an input of energy to do that? Yes. Is there energy stored in the fact that the electron is further away? If we let it drop back down, we get that energy back again, right? So these excited states represent a pigment molecule that has an electron that's been pushed out to a more distant orbital through the absorption of a photon. Let's look at this in a diagram that will help us see what's going on here. So we call the lowest energy state of a pigment its ground state. And then there may be a number of different excited states. And those excited states correspond to the absorption of a photon with a certain energy in it. So on the y-axis here, we've got energy, right? So the absorption of a red photon by chlorophyll raises the molecule from the ground state to a low energy excited state. The absorption of a blue photon raises the molecule from the ground state to a higher energy excited state. It's kicked that electron further out. In both cases, the energy of the photon has been conserved in these excited states. So the next thing we need to think about is how is this energy that's stored in the excited state used in photosynthesis? Okay, that's the key here. So let's think about, first of all, about the differences between the blue excited state, the thing that counts for the blue peak in the absorption spectrum. Here's the absorption spectrum of chlorophyll with a big peak in the blue and a big peak in the red and not much in between. So if we absorb up to the blue excited state, in the first approximation you might think, whoa, there's almost twice as much energy in a blue photon as in a red photon. Can you do more photosynthesis in blue light than you can in red light? If you didn't know anything else about photosynthesis, but I told you there's twice as much energy, would you think that you could do more photosynthesis with blue light than with red light? Yeah, it makes sense. But you can't. And the reason you can't is due to the photophysical properties of not just chlorophyll, but any excited state. These higher level excited states are extremely unstable. The lifetime of that excited state is about 10 to the minus 14th seconds. 10 femtoseconds. That's not very long. And what limits the lifetime is that energy can be given up as heat through molecular vibrations very quickly. This is true not just for chlorophylls, it's true for virtually any molecule that absorbs light energy. The higher level excited states are unstable, and they decay very quickly by giving the energy off as heat down to the lowest excited state, corresponding to the red absorption. So all that extra energy of a blue photon is lost instantly as heat, immediately after absorption. Nothing else is fast enough to compete with that. So the short answer is no. You cannot do any more photosynthesis with blue light than red light. The only thing that happens is the plant heats up more. Okay? And again, this loss of energy between these two excited states is a common property of all molecules. Biology can't alter that. It's a fundamental property of the way molecules work. Yes?
So what happens with the light between blue and red? Why is absorption so low?
Why is the absorption so low?
Yeah.
Okay, so remember that in the process of absorbing the photon, the energy of the photon has to show up as the energy of the excited state. In other words, there has to be conservation of energy. So these excited states, the energy of these are fixed by the properties of the molecule. So if that energy match has to happen, if you have a red photon, there's a perfect energy match. Absorption is very common. In the green part of the spectrum, there's no excited states there. So even though you put green photons with intermediate energy in there, they're not absorbed because there's no state that has an energy match. So you just learned some very fundamental things in quantum mechanics. The reason that molecules absorb light at specific wavelengths and not others is because there has to be excited states whose energy matches the energy of the photon in order for that absorption to occur. If there's no excited state there, there's no absorption. So the fact that chlorophylls don't absorb anything in the middle part of the spectrum, in the green and yellow part of the spectrum, is a consequence of the fact that there's no excited states with corresponding energies, with equal energies to those photons. Okay, so this in some ways makes photosynthesis a whole lot easier, because what that means is everything in photosynthesis proceeds from this lowest energy excited state. So we just need to think about the properties of this lowest energy excited state to see what's going on. So let's ask the questions, what are the possible fates of this excited state? Well, in photosynthesis, we know what we'd like to do is use this energy to drive redox reactions, to move electrons around, to make electron transport work. And we'll see the details of that in just a second. That doesn't happen if you take pure chlorophyll in solution. Take the same molecules that are functioning in photosynthesis in the thylakoid membranes of a chloroplast and take them out of the proteins and put them in solution. They can't do photosynthesis. Those excited states will decay back down to the ground state by processes that are built into the chlorophylls. One of those is to give up the energy as heat, just like it did up above. It's just a lot slower. This excited state will stay around for 10 to the minus 8th seconds. Still pretty fast, but a lifetime compared to how long those excited states hang around. So the blue one, 10 to the minus 14th seconds. This one, if you put the chlorophyll in solution with no proteins around it, it would be about 10 to the minus 8th seconds. So a lot longer. So this excited state can give up its energy as heat. It can also give up its energy in a process called fluorescence. Absorption took the energy of a photon and created an excited state. Fluorescence is the opposite reaction, where an excited state gives up its energy by creating a photon. And if you haven't seen fluorescence, I think I got a picture here, yeah. So here is a test tube with chlorophyll in it, a blue light source, which is creating excited states. And remember, those blue excited states decay back down to the red excited state. And then here is the chlorophyll going from its first excited state, its lowest excited state, back down to the ground state by giving off red photons. So this is another possible mechanism by which this lowest excited state can give up its energy back down to the ground state by emitting fluorescence. And in fact, the majority of work that goes on in my laboratory to study how photosynthesis works is looking at characteristics of this fluorescence, because it tells us a lot about what's going on. Francisca?
Can you look at that picture from before? Because theoretically the next chlorophyll absorbs the red photon.
Sure. So what you're asking here is, so there's one chlorophyll molecule right here, right? And it gives up a red photon. Could the neighboring chlorophyll molecule absorb that? Absolutely. It turns out that the probability of that, of fluorescence is relatively low, maybe 20%. And the probability of it being reabsorbed, well, it depends on how dense the chlorophyll solution is.
But the solution, it would be fairly higher compared to living in a
So are you getting at, could emission, fluorescence emission, reabsorption, fluorescence emission, reabsorption be a mechanism of moving excited states from one chlorophyll to another?
Well, theoretically, yes, that is so unlikely to happen. But in that solution, it could theoretically happen.
Yes.
I have not, maybe not a long chain, but maybe from one to the next, and then.
Yeah, you can take, the answer to that question is relatively easy to assess. If you take a solution, a dilute solution of chlorophyll, and shine blue light on it, this is what you'll see. If you take a concentrated solution, same molecule, and shine the blue light on it, you won't see any fluorescence. And the reason you won't see any fluorescence is because you've increased the number of neighboring molecules that could absorb that red photon. So none of the light gets back out again. Okay, but what you're saying is correct. Okay, one more thing we need to think about. It's not on this diagram, but it's extremely important. It'll come up several times when we talk about photosynthesis. Another way this excited state can decay is through what's called a triplet state. So if you remember from chemistry, something about triplet states are where the electrons are not anti-parallel, but they're paired. It's not important that you understand that. But chlorophyll triplet states these excited states are really bad. Remember, did I talk with you last time about chlorophyll being one of the most dangerous biological molecules known? I mentioned that? Yeah, okay. This is why. Because these triplet states react with molecular oxygen to produce the excited state of oxygen, called singlet oxygen. And this is toxic to cells. It causes, for example, immediate damage to any lipid that it comes in contact with. So the excited states of chlorophyll in the presence of oxygen can sensitize what we generally refer to as ROS, reactive oxygen species. And we'll see 2 lectures from now when we talk about the ecology of photosynthesis that this is really important. And when we talk about stress physiology at the end of the semester, these will turn out to be very important as well. Okay, so we've talked about what chlorophyll does when it's out on its own, but we still haven't talked about how this excited state energy is used to drive photosynthesis. So that's what we need to finish up with today, and obviously we won't get through all of it, but we'll pick up the end of it next week. Okay, so one of the things that is interesting to think about is what is it that makes a molecule be able to absorb light energy easily? There are some molecules that absorb light energy easily and some that don't. For example, sugars, sucrose, does not absorb light energy easily. But the red pigments in Mike's sweatshirt, right? Or the black ones in Anna's sweatshirt or whatever, those pigments are absorbing light because they're giving rise to the colors that you see. What are the properties of molecules that give rise to strong light absorption? And the main thing is systems of alternating single and double bonds between carbon atoms. So here's a molecule called carotenoids. Beta-carotene is one of the most common ones, the things that makes carrots orange. Here's a chlorophyll molecule, and you see in the chlorophyll molecule patterns of alternating double and single bonds. Those molecules that have lots of those tend to be very strong absorbers of visible light. Okay. Chlorophylls and carotenoids aren't the only molecules that are strong absorbers, but they're the ones that photosynthesis evolution is selected for to function in photosynthesis. Okay, so what I want to do is switch gears and this will probably be the last thing we do today, is to talk about 2 key experiments that were done back in the 30s, actually, 1930s, so a while ago, that gave us a tremendous amount of information about how chlorophyll functions in photosynthesis and what the reactions are that are going on there. And these were done by 2 scientists, Robert Emerson and Daniel Arnold. Emerson was head of the lab, and Arnold was one of his graduate students. And one of the things that had been invented in the 30s that turned out to be an extremely useful tool in photosynthesis was essentially the equivalent of a flash bulb. Now, you guys are probably all too young to remember when you actually had a lamp that you put in the camera that you used it once and it made a flash, and then you take it out and put another one in. But those flash bulbs were invented back in the late 1920s. And the thing that was nice about it was they produced a very narrow pulse of light. That is, the light is on, so this represents the light off, the light on, and the width of this was on the order of 1 to 5 microseconds. So 1 times 10 to the minus 6 seconds. A very short and also very bright pulse of light. And what Emerson and Arnold did was made use of this to study the reactions in photosynthesis. And here was their rationale. If we use very short pulses of light, separated by long dark periods, then we can isolate one set of reactions that happens when you give that pulse of light, and then let everything else that's happening after that get over with, and then give it another pulse of light, and study, for example, saturation effects. So their idea was to have a pulse of light, each of these pulses of light being on the order of 1 microsecond width, and then the time between the pulses being about 0.1 seconds. So very short pulses of light separated by much longer periods. In fact, they did a lot of experiments to find out what was the sort of the optimum time between these pulses. And it was on the order of a couple tenths of a second. Okay, so we give a short pulse of light, we wait a long time, we give another pulse of light. And in this experiment, what they did, so this is the light end of things, and they also measured O2 production. So when you gave a pulse of light, what they saw was oxygen being produced. And this sort of long tail there represents not how long it takes the oxygen to be produced, but how long it takes the electrode that they're using to measure oxygen to detect it. So they give another pulse and they'd see more oxygen. And all they did was they integrated the area underneath this curve associated with each pulse. And they made a plot of the intensity of the pulses versus the amount of oxygen that's produced. Pretty straightforward. It's just like an enzyme experiment. Light is the substrate, oxygen is the product. So not surprisingly, they saw a light saturation effect. But what was surprising was their interpretation of what this experiment means. So first of all, if we think about the initial slope of this curve, the slope is the change in y divided by the change in x. Oxygen produced per photon that was absorbed. And they did something very clever that had never been done before. They can't measure how many photons were absorbed very well. They can measure how many photons were produced by their flash lamp, but they can't measure how many were absorbed. So what they did was they used algae, single-celled algae, and they made a very dense culture so that every photon that hit the culture was absorbed. Right? So basically all the light that entered into the culture of algae was absorbed. So they knew how many photons were absorbed, and they knew how much oxygen was produced. The slope of this line is oxygen per photon. Right? So what they found was for each oxygen produced, it required the absorption of 9 to 10 photons. This is called, they got this backwards in the textbook. This is not the quantum yield. This is the quantum requirement. It is photons per oxygen. It's telling you how many photons are required to produce one molecule of oxygen. Okay? This was completely new information. Nobody had any idea. Everyone assumed it was 1 photon per oxygen or 1 photon per CO2 fixed. The fact that it was 9 to 10 was very surprising and tells us there's a fairly complicated mechanism that's going on there. The other thing that was really surprising was the maximum yield. So what they calculated was, what's the highest rate of oxygen evolution that they measured at saturation? And then they said, okay, we know how many total chlorophylls there are in the solution. And they just took a ratio. At the maximum rate of photosynthesis, how many chlorophylls are there per oxygen produced? Or other way around, oxygens per chlorophyll molecule. And they found 1 oxygen per 2,500 chlorophyll molecules. The expectation was that that would be 1 oxygen per chlorophyll, that each chlorophyll did this reaction on its own. This was a totally shocking conclusion, but it was the first indication that in photosynthesis, chlorophylls don't work by themselves, that they work in units together, where lots of chlorophylls function as a unit to facilitate these reactions. Okay, so 2 important things. This tells us that it takes many photons per oxygen produced. And it also tells us that for each oxygen produced at the maximum rate, it takes the cooperation of a lot of chlorophylls. Very key observations that once we think about the Z scheme and all the reactions of photosynthesis, these make a lot of sense. So we'll come back to that in just a second. Okay, second experiment. Also done by Emerson. Relatively simple experiment again. He's measuring rate of photosynthesis, and he was also measuring it by oxygen evolution, oxygen production. And he did this with 2 different, not just 2, he used many different colors of light. But what he found that was most interesting was when he used red light being wavelengths that were less than 680 nanometers, and far red light wavelengths that were greater than 680 nanometers. So what he did was he illuminated his algal culture using the same chlorella that he used for the previous experiment with low intensity far red light. Turn the light on. So this is now tens of seconds here. Turn the light on. You see oxygen evolution, a certain amount of oxygen evolution. Turn the light off. The oxygen evolution stops. Now turn on a low intensity red light. And you see some oxygen evolution. He actually adjusted the intensities of the red and far red, so the amount of oxygen evolution in those two wavelengths of light was about the same. Then what he did was he turned both the red and the far red on at the same time. And the expectation was that what you would see would be something that would be the sum of these 2. And what he saw was something that was much greater than the sum of the 2. He referred to this effect as enhancement. That there is a synergy between the red and far red light. That something more happens in red plus far red light that can't happen in either one of them alone. And this was the first indication that among that, in the experiment he did previously, where there was 2,500 chlorophylls that were interacting with each other to give rise to 1 molecule of oxygen, that those 2,500 chlorophylls are not homogeneous. That there are 2 separate pools in there. One of those pools of chlorophyll absorbs at wavelengths less than or greater than 680 nanometers, and the other absorbs at less than 680 nanometers. And either one of those alone is moderately effective at driving photosynthesis. But when you put them together, there's a much greater effectiveness. And this was the first suggestion that, as I said, there's 2 different pools of chlorophylls. And we know now, and Emerson suggested it first, wavelengths that are absorbed at greater than 680 nanometers are preferentially absorbed by photosystem 1. He called this photosystem 1. And the wavelengths of light that are less than 680 nanometers are preferentially absorbed by photosystem 2. And his suggestion was that PS1 and PS2 somehow must function together to carry out the reactions of photosynthesis, not just one or the other. And we know now that photosystem 1 and photosystem 2 function in series. Okay, we're going to leave off here, and we'll pick this up next time. We've actually got the next 3 lectures are related to various aspects of photosynthesis, so we'll sort of carry over among them. We'll pick up, we'll get caught up by the end of it.